All right, welcome to The Explainer. Today we are getting into a really interesting one, Paracoccus yeei. This is an organism that's kind of crept out from the fringes of microbiology and landed squarely in the clinical spotlight. So if you're working on the front lines in a diagnostic lab, you're going to want to hear this.
So, just so you know where all this is coming from, we put this explainer together using a pretty comprehensive Gemini deep search of all the latest research. And hey, if you want to nerd out and go even deeper, we've got you covered. All the primary sources and citations are right there for you in the linked notebook document.
Okay, so here's the plan. First, we'll talk about how this bug came out of nowhere. Then we'll get into its really unique genetics, what it actually does to patients, how you can spot it in the lab, why treating it is so tricky, and finally, what we can do to prevent it and what's next.
All right, let's kick things off with the backstory. You know, for years, Paracoccus yeei wasn't even called that. It was basically a mystery organism, just known by this cryptic code name E02. We're going to look at how modern lab tech finally dragged it out of the shadows.
So for the longest time, this thing was just getting missed. It was picky about how it grew, and it looked a lot like common skin bacteria, so you can see how it'd be overlooked. But then, technology changed everything. The widespread use of stuff like MALDI-TOF MS and 16S rRNA sequencing has been a total game changer. We can finally see this organism for what it is.
Okay, next up, let's get into where Paracoccus yeei fits in the grand bacterial family tree. And this is the really cool part. We'll look at what makes its genome such a fantastic example of a pathogen literally building itself as it goes.
Okay, so on paper, it's a gram-negative bacterium alpha proteobacteria class, standard stuff. But here's the kicker, and this is really important. Its genus, Paracoccus, has over 80 species, most of them just hanging out in the environment. Paracoccus yeei is the only one that's consistently tied to human infections. So that begs the question, right? What makes this one so special?
Well, the secret is in its genome, which is just incredibly dynamic. See, most bacteria just have one main chromosome, their single instruction manual. Not this one. Paracoccus yeei has what's called a multipartite genome. Think of it like a main book plus a whole bunch of pamphlets and sticky notes. In fact, more than a quarter of its entire genetic code is on these smaller, mobile pieces.
And that brings us right to the heart of what makes it a pathogen. This collection of mobile genetic bits and pieces, its mobilome, is like a library card for genes. It can just borrow them from other bacteria. So its toolkit for causing disease isn't something it was born with, it's assembled. It's a patchwork of borrowed powers pieced together through horizontal gene transfer.
And this borrowed toolkit has some seriously impressive gear. For starters, it's got a unique urease gene cluster. And remember that, because it's a huge clue in the lab. It's also got these things called type 4 secretion systems, which are like tiny molecular syringes for injecting stuff into our cells. And, to top it off, it has powerful defenses to shut down the attacks from our own immune system.
Okay, so let's switch gears to the clinical side. Who gets sick from this bug, and what does it actually do to them? This is the stuff you absolutely need to know when you're staring at a potential isolate on your bench.
Let's be clear, this is a textbook opportunist. You're just not going to find Paracoccus yeei causing trouble in a healthy person. No way. It almost exclusively goes after patients who are already in a tough spot, people with severely compromised immune systems from things like cirrhosis or cancer, or, and this is a big one, patients with indwelling medical devices. Those catheters are like a perfect landing strip for it to set up shop.
And the types of infections it causes are pretty specific. The number one thing you'll see is peritonitis in patients on peritoneal dialysis. That's the most common by a long shot. It can also get into the bloodstream, causing bacteremia, but often without that full-blown septic shock you might expect. And then there's this weird thing it does. It seems to have a real liking for eye tissue, causing nasty cases of conjunctivitis and keratitis. And just to show how serious it can be, while it's super rare, it has been found causing devastating meningitis in fragile newborns.
So, this brings us to the million-dollar question, right? How do you actually pin this thing down in the lab? This is where you put on your detective hat, because there are some very specific clues to follow and some tricky traps you'll need to avoid.
Your first big clue, and it's a pretty distinctive one, can come right from the gram stain itself, especially if you're looking at the gunk from a positive blood culture bottle. Keep your eyes peeled for these really unique, hollowed-out-looking cells. They've been called O-shaped or donut-shaped. Seriously, if you see that, Paracoccus yeei needs to jump right to the top of your list. But here are the two biggest traps you can fall into. First, that gram stain can be a real liar. The cells can look kind of gram variable, making you think you're looking at something else entirely. And second, and this is a huge one, after 24 hours, the colonies look exactly like your run-of-the-mill coag-negative staph, just small white non-hemolytic dots. It is so easy to just write them off as contamination. And that's exactly why you need a system. Start with a really careful look at that gram stain. If anything looks even a little bit weird, do not just toss the culture. Give those plates a full 48 to 72 hours. You're looking for them to get kind of slimy and mucoid. Then hit it with your key biochemicals. It should be oxidase positive, catalase positive, and here's that key marker again, urease positive. Then, of course, lock it in with MALDI-TOF or 16S sequencing for the final definitive answer.
So here's a quick cheat sheet to help you tell it apart from other usual suspects on the bench. That positive oxidase test, boom, that immediately rules out Acinetobacter. Now, Acinetobacter can also be urease positive, just like Paracoccus yeei. But the key combo to look for is oxidase and urease positive. That really helps you separate it from something like Moraxella, which is going to be urease negative.
Okay, let's talk about what is probably the biggest clinical headache: treatment. You've done the hard work, you've identified Paracoccus yeei. Now what? How do you actually guide therapy for this thing?
And right away, we run into a major problem. A huge one, actually. Neither EUCST nor CLSI have any official specific breakpoints for Paracoccus yeei. None. So that leaves all of us in this massive gray area, trying to interpret susceptibility results without any real rulebook to follow.
So what's the workaround? Well, the current thinking is to very cautiously borrow the breakpoints for Pseudomonas aeruginosa. Look, it is not ideal, not by a long shot, but it's the best we've got for now. And based on that, it seems to be generally susceptible to carbapenems and piptazo. But, and this is a big but, when it comes to third-gen cephs and fluoroquinolones, it's all over the place. You really can't count on them working.
And this just shows you how big the data gap really is. There are 0 specific treatment guidelines from the big international groups like ESCMID or IDSA. The Dutch guidelines from SWAB, nothing specific there either. The basic advice is just treat based on the patient and your local lab results. And what about kids? It's even worse. Our entire playbook for treating children comes from a single case report. 1. A neonate with meningitis who was treated with meropenem and amikacin. That's it.
All right, let's wrap this up by talking about prevention and what needs to happen next. Since this bug comes from the environment, how do we keep it away from our most vulnerable patients?
When it comes to prevention, it's really all about mastering the basics. This bug comes from soil and water, and we know it can hang around on surfaces in the hospital. So our strategy has to hit it from all sides. That means relentless hand hygiene, really thorough cleaning of the environment, keeping an eye on hospital water systems as a potential source, and, this is absolutely critical, using strict aseptic technique every single time you're dealing with a line or a catheter.
So the path forward is pretty clear, right? We urgently need official AST breakpoints, and we need to start building clinical registries so we can have some real data to work with. But that leaves us with one last thought, and it's a bit unsettling. Remember that assembled genome? Its greatest trick is its ability to learn from other bacteria. So the real question is, what's it going to learn next? What new weapons is it going to pick up?
Hey, if you found this deep dive useful, do us a favor and subscribe to the channel. We've got a lot more clinically relevant micro coming your way.
And we really want to hear from you. Have you ever come across Paracoccus yeei in your lab? Did you ever spot those weird O-shaped cells? Drop a comment below and share your story. We'd love to read it.